Okay, I think we are 20 participants. It's okay. So I start introducing Francesco Torricella, today's speaker. So Francesco is now a postdoc at NIH, and he's a chemist. He did his studies in Italy, both the bachelor in chemistry in University of Camerino and the master in chemistry when he moved to Florence, working at CERM with Lucia Banci. During this time, he spent a year or so in Göttingen, where I met him, and he worked in Marina Bennati's lab doing pulse CPR and distant measurements. So there he learned DEER, and this is what he's still doing at NIH. After his during the PhD, he went also to NIH, to Mario Sclorer lab. He had a pre-doctoral fellowship to stay there for a couple of years. I think he arrived during the pandemic, right on time. And there is, during the PhD, he did mostly in-cell EPR and liquid state NMR, looking at 19F nuclei. So during this time, he developed a method, a protocol, that is going to talk about it today. And this protocol is to basically deliver proteins inside cells with a mild thermal stimulation. And after that, you can study this protein with CWPR and DEER. So yeah, this is the topic of the talk. And so Francesco, you can go ahead.
All right, thanks. Thanks, Thomas, for the really kind introduction. And yeah, well, as Thomas already said, I'm going to be talking about one of the projects that I did during the PhD when I was back in Florence. So just to start out the presentation, I would like just to give you like really a really quick outline of what I'm going to show you. And so the entire presentation is going to be focused on these two, mainly on these two articles, which we managed to publish during the previous years. And yeah, and the first one is going to be focused more or less on the bacteria and yeast cells as cell host for our in-cell experiment. And the second one is going to be mainly focused on the mammalian cells. So just to give you some introduction about the in-cell idea, well, even if we already had the chance probably to listen to some in-cell presentation in this meeting seminars, well, I thought to give you like a flavor of the methods that are already available to prepare in-cell samples for either in-cell NMR experiment or in-cell EPR experiment. And if you want to start out to run this kind of experiment, which are basically defined as this branch of approaches where the main idea is to look at the protein that you are targeting, that you want to study. The idea is to look at this protein directly inside the living system, and not anymore to look at them directly in the buffer solution. So if you go on literature and you search for this, you're going to end up with two main approaches for sample preparation, which is actually the bottleneck of these approaches. And you will see that there is the first branch of approach, which are called protein overexpression, in which basically the main idea is really, let's say, I would say easy, especially for the one who in general express protein, because the idea behind the overexpression is to deliver the DNA inside the host that you selected, which can be like bacterial cells, yeast cells, insect cells, or mammalian cells. And once you deliver the DNA, what you can do is basically you can start the expression of this heterologous DNA using specific molecules, and you're going to end up with some cells that are really full of the protein that you selected. And if you do all of this procedure in a minimal media where you label the media, you can easily understand how these approaches can be applied to in-cell NMR. For in-cell APR, this kind of this group of protocols can still be applied with the latest results published by the group of Drescher. But let's say that those are not the most frequent ones. On the other side, if you go deeper in literature, you will see that there is this other, let's say, phase of the medal, where the idea is not anymore to deliver the DNA, but instead of this, the idea is to deliver directly the protein, which is already purified and for in-cell EPR purposes is already labeled with some paramagnetic labels. And if you search on literature, you're going to see that the main methods are represented here. So the most famous one is the electroporation, which, well, it has been used a lot, like with remarkable contributions from the group of Daniela Wolfart. And then there is this CPP delivery, which stands for cell-penetrating peptide delivery. And then there is the pore-forming toxins and the microinjector in the injection in the oocytes, which is probably the easiest one from theoretical point of view. So going through the idea that I got when I was back in Germany, and then I managed to realize this when I went back to Italy to start the PhD. So whenever we start to prepare proteins, what we normally do is to transform our bacterial cells with some DNA, and the DNA is encoding for the protein that then we are gonna purify, and we are gonna use it for our experiment. So the idea here is basically that during this transformation protocol, what you are doing is basically a delivery. You are delivering the DNA directly inside the cell. And that was the starting point, because what I was thinking at the beginning was, when I was still not in this molecular biology field, was why to not use directly protein instead of DNA, but keeping more or less the same protocol. That was the idea. And at the beginning, I was pretty shy on this, because if you think on it, well, it's really easy. It's something that, well, you can say, well, probably someone else did it before. But then I spent like a lot of time in literature searching for some references, and nothing was there. So we started to do some experiment. So before starting the experiment, obviously we set up, we managed to build up like a protocol, 3-step protocol, really easy. It's really, for the ones that are working with proteins, this is more or less the transformation protocol. And what we did at the beginning to check if the protocol was working towards the delivery of the external protein, we used some fluorescence protein. And for this reason, we used 2 selected proteins. The first one is the famous EGFP protein. We use the EGFP, which is a little bit more fluorescence, let's say. And we decided to use the ubiquitin with a specific point mutation, the A28C, and we labeled this cysteine with a fluorophore, which was the atomalamide 4A. And as you can see, well, it's not that clear from these images because we were close to the limit of our confocal system, but you can more or less appreciate how the protein is delivered inside the cells, and you can get the fluorescence, you can understand where the cells are, and you can more or less understand the shape of this E. coli, which are really small and which is way more, which is a lot complicated to see them at the confocal side. So on the same line, what we decided to do at the beginning, since I was coming from Germany, I was coming from this from the Bernatis group. The idea was to use EPR because I was interested in using this kind of spectroscopy. And for doing that, what we decided to do was to select several proteins to show how this protocol can work with different types of protein in terms of shape and structural and secondary structure. And this is what we got at the end. Basically, we decided to use ubiquitin, the BIF3 domain, which is a small domain of the CIAP protein, which is a human protein, really important for the cancer regulation. Then we use delta FUS, which is again is a domain for this fusedine sarcoma protein, which is still a human protein. Then we selected the EGFP for the confocal story, and the carbonic anhydrase 2 because it was globular, it's quite nice as a protein, and it has like a natural cysteine there. So what we did at the beginning was to select a spin label. We decided to use the maleimide proxyl, which is known to be non-stable upon reduction, but we decided to go ahead, even taking into account this. And what we did was to label the proteins, and those are more or less the spin efficiency that we got. And as you can clearly see, on the case of carbonic anhydrase 2, the yield was, the label fraction was pretty low. And the reason for that is simply because the cysteine is really buried in the structure of the CA2, but it's still a little bit accessible. So we were getting like spin labeling efficiency in the range of 10%, 7%. Those were the values. So right after that, what we did was to firstly record the in vitro experiment. So we recorded the spectra at the CW EPR, at the expand, at room temperature for the proteins in buffer solution. And those are reported here in black. And right after that, we use those proteins at a concentration of 1 millimolar external protein concentration, and we recorded the in cell. In this line, on this line, it's really important right after the delivery to be sure that whatever is outside the cells, it must be removed, because otherwise you're going to get trouble with the understanding of your final in cell experiment. And well, on this specific case with the in cell experiment in bacteria, and as I will show you even on the other cell situation, what we saw is what I said previously. So this kind of maleimide proxyl is not stable upon reduction, but and you can clearly see looking at the intensity of the signal over time after the delivery. So basically, if you look at the signal over time in your in-cell sample at room temperature, the signal is going down. Why is going down? Is because the diamagnetic product of the nitroxide is not it's diamagnetic, so it's not going to give you signals, and that's what is going on in cell. So basically, your nitroxide is getting reduced, and you're going to lose your signal really quickly. So there are so many ways right now in literature for, let's say, more or less overcome this problem, like using shielded nitroxide or tritium radical or metal-based label. So after this first section on bacteria, what we decided to do was to move to some more interesting systems, which we selected to be Pichia pastori cells. Those cells are really famous for protein production in yeast, and that's why probably I found them in Florence. Well, the idea was, okay, let's move now. Let's try to use the same approach to re-optimize again the protocol, but using some more interesting cells, which are like eukaryotic cells. And this is what we end up with. So as you can see, it's quite easy. It's like 4-step protocol. And after that, what you can get is this. So if you deliver EGFP or ubiquitin labeled with this fluorophore, you're going to see that the protein is well spread inside the intracellular environment of those cells. And there is one really interesting stuff that I want to show you. If you look at this picture here, well, let me, okay. Okay. I have no idea how to get my pointer, but I'm gonna try to tell you this. So if you look at the first panel, the time 0, you're gonna see that some cells are budding. And the reason why they are doing that is because this Pichia pastoris is a budding yeast. So whenever they are replicating, they are budding. And the interesting feature is that the protein is going from the first cells to the second one, to the newborn. And as you can see, the green fluorescence is like is going from the old cells to the new cells. And this is quite interesting, even because it's telling you that the method is not affecting the physiological condition of those cells if they are still able to replicate even after the delivery. So on the same line, we went farther ahead. We used the same protein for the in-cell CWEPR experiment. So we delivered the cells inside this Pichia pastoris. We delivered the protein inside this Pichia pastoris cells, and we got these results again. Well, you can see the in vitro, which is more or less the same as before. But in this case, well, some of the spectra were quite different power. And then we moved to the in-cell condition, and well, we got our CWEPR spectra. Again, even in this case, we saw the same behavior for the myeloid proxy, and we saw the signal going down over time, which is, from some perspective, is telling you that the protein is well delivered inside cells. If you see this behavior, that means that if you are sure that you wash your cells like properly and nothing is outside and all the dead cells are gone, are washed out, you can use this as a double check of your delivery, the fact that your signal is going down. Okay, well, in the meantime, in conjunction with this in-cell EPR experiment, what we thought was really important was to establish how much protein was going inside the cells. And we did that with 3 different methods. We used Western blot, which is a proper biological stuff. Then we use in-gel fluorescence page, which is, again, it's more or less an SDS page. And then we use the double integral of the CWEPR spectra for getting a rough estimation of the concentration. And we end up with those values. So just to give you like a rough idea of the mean concentration that we were able to get, it was more or less around like 3 micromolar, 5 micromolar. Sometimes it was higher, and sometimes it was less. So in mean, well, the mean concentration is around 2 micromolar, 3 micromolar with this setup. So going back to the CW experiment, what we did on the article, and you can see that on the article, is really it was to simulate those spectra to see the effect of the in-cell environment to the line width or to the dynamic of the protein upon the delivery. And yeah, well, basically we did that systematically for all experiment, and you can see this table even on the paper where we compare the in vitro case, which was mainly a single component experiment, single component curve, to the in-cell one, which are more close to be double component, and which is probably the effect induced by the intracellular environment. So right after this first part of the work, which was published as an article as it is, without using their spectroscopy, because we were more interested in saying, okay, well, this is a new method that everyone can use for doing in-cell EPR. So we use model proteins, so it's not that interesting to go farther in year or something like that. So we just closed that story and we opened a new one. And this new one was, as you probably already got, it was close to the fact that we use bacteria, we use yeast, and now it's time to use mammalian cells, which are known to be really fragile, which are known to be more tricky to work with, and all of these other problems with the handling and whatever. But we tried to do that. The idea was the same, to use the same approach, to use thermal treatment as delivery method, and to use another cell host, which happened to be mammalian cell. And basically, we came up with this protocol. Well, I can show you here. We came up with this protocol where using adherent cells, the first step is to detach the cells. Once you detach the cells, you can resuspend your pellet cells with the proteins at the concentration that you want to use for the delivery. And then there's the proper thermal treatment, which is composed by several treatment at 42 degrees for 5 to 10 minutes, and well, intercalated by 2 minutes in ice bath between each temperature shock. So the first stuff that we did, since those cells are known to be really fragile, we checked the stability of the cells in terms of viability. So what we did was we did like multiple experiments where we were testing just the shocks. Well, we used three different approaches, three different setups, sorry. The first one is called 5-5, and which stands for 5 minutes at 42 degrees. Then the dash is 2 minutes in ice, and then 5 minutes is again 5 minutes at 42 degrees. And at the end, there is another ice bath. And so basically, as you can see here on the x-axis, we use three different methods. We use 5-5, and then 5-5-5, and then 5-10. And from the viability assay that we ran, we basically saw that the viability was not that changed compared to the control, which is a really good stuff, because again, those cells are sometimes really fragile. And so we were really happy with this. And we did that with HEC-293 cells, HeLa cells, and URCA-T cells. These first two cell lines are like attached cell lines, so they do grow on adherent flask. And the URCA-T, since those are lymphocytes, those are like living on suspension. In the case of URCA-T, as you can see, the 5-10 protocol was not working that cool. So that was the reason why we removed that. And we went ahead just with 5-5 and 5-5-5 in the case of URCA. Right after this first test, what we did was to test the viability with the protein. Well, in the sense that we took the cells, we took the protein, we mixed them like in the proper protocol, and then we tested the viability using just one single delivery method, which was the one that we used for all the other measurements. We was 5-5 delivery method. And as you can see, again, the viability is really high compared to other like treatments. These values of viability are really good, and the values are high even for high external protein concentration. As you can see here on the x-axis, we tested the viability upon the use of an external protein concentration of 1 millimolar, 0.5 millimolar, and 0.1 millimolar, which happened to be the concentration that we selected for our experiment. Last biological stuff. So in addition to this viability test, what we decided to do was to see how the expression of some heat shock proteins, which are reporter of this heat shock, actually, was changing upon our delivery method. And well, as you can see here from this Western blot against the HSP 70s, the delivery is not affecting more or less at all the overexpression, the expression of the basal HSP70s, which is again a really good story, because at the beginning we didn't know how how exposing the cells at 42 degrees can affect, or it would have been effective towards the expression of this kind of HSPs proteins. So yeah, after the setup of the protocol, again we went ahead with confocal microscopy. All these images have been acquired in non-adherent conditions. So it means that we did the delivery and we do not reattach the cells, those that are living in adherent condition. And so that's why they seem to be globular. And as you can see, well, you can understand where the protein is more or less. You cannot distinguish in this case if the protein is in the nucleus or not, because again, those are not reattached cells, so it's way more trickier, it's way more complicated to say that. But what you can understand is that the protein is inside, it's delivered, and you can extract some concentration out of this. After the delivery, what you can do is basically to lysate your cells, run some Western or in-gel fluorescence space, and you can get those concentration values. And here, basically, the panel A is referred to the concentration that we got for ubiquitin, labeled with atomistic, delivered with different external concentration and with the different protocols, with different methods, delivery method. And on the other panel, on panel B, what you can see is the same story, but for the GFP. In the case of GFP, we use Western blot to get the concentration, and in the case of ubiquitin, we use in-gel fluorescence phage. Same story, but less protein. So in the case of this this work, we selected just the ubiquitin since it was way more easier, let's say, to work with just one protein, and then well, someone else, or we can extend more the limit, the upper and the limit bound of the applicability of this method. We basically in this story we use just ubiquitin protein with two different mutations. The first one for CW-EPR spectroscopy, we selected the ubiquitin A28C and we label it with the mammalian peroxide. We got the experiment, which are the gray one here. Those are still room temperature experiment, X-band, nothing really fancy. And again, we saw the intensity, the decay of the intensity over time, as you can see here in this plot. And more or less the lifetime of this nitroxide is more or less the same between bacteria, yeast, and mammalian cells, which makes sense because, well, those environments are really full of reducting agents. Yeah. So after this, what we did, as I was saying before, we selected another mutation for the ubiquitin. Basically, we decided to use the S20G35C ubiquitin, and we double labeled the protein. And basically, we set up everything for running some positive EPR experiment, which are way more interesting than CW-EPR. So as you can clearly see here in this panel, well, the section A is just the protein, the ubiquitin, doubly labeled with this mammalian peroxide. The model is coming from MMM, and those are like just the rotamer library is just for description. So in panel B, what you can see is the decay of the intensity of the nitroxide intensity in the case of the double label ubiquitin delivered inside egg, pure cut and HeLa cells at room temperature and at X-band. And the asterisk basically is giving you the position where we more or less we snap freeze our sample, like right after the dead time, the preparation dead time of the sample, which is more or less 10 minutes, well, a little bit more. It's like 11, 12 minutes. We snap freeze our sample, and the sample was brought to the Q-band machine. And those are the data that we got from the Q-band. So in panel C, you can see the ecofilm sweep that we recorded. The blue shaded area, of course, is the nitroxide signal. And in panel D, what you can see is basically the experiment that we recorded for the deer experiment that we got for the in-cell sample. And even here, you can realize how the overall concentration, delivered concentration, is not stable. It's changing upon cell lines, and it can be really variable. Like in the case of HeLa cells, you can see that, well, the intensity of the signal is way more, it's way better than the one that we got in X293 cell. In the panel D, again, the blue spectra is just the deer experiment that we recorded in vitro. So it's like 60 micromolar protein concentration. And yeah, and the cyan one is the in-cell one. So going ahead, what we decided to do at the same time was to select another label, which happened to be the maleimide DOTA gadolinium, which is basically stable upon reduction because it's a metallic one, is a metal-based one, so it's stable, as even like all the Daniela's work have shown. So we double labeled the ubiquitin with this maleimide DOTA, and we run the same delivery and we run the same experiment. In this case, you can basically appreciate, well, those are just rough estimation of the concentration, and on the same panel, you can understand, well, you can see the ecofilm sweep experiment at the Q band at 10 Kelvin, and the deer experiment that are coming from our sample. So the red one is the in-cell experiment, and the blue one is our in vitro reference. So with all this deer traces, what we did was to analyze them with 3 different methods. We used DeerLab BD and WAVEX BD. And why we did that? Because we basically, well, I thought to use different methods to have in order to retrieve something like an error in the distance estimation. And what we end up with was is represented here. Basically, you can clearly see the mean distance that we extracted for this protein, for the ubiquitin delivered in vitro and in the in-cell condition. And that's already known. Well, this protein is known to be like a stone, so it's not changing a lot upon the delivery. So it's not changing the distance of this specific mutant. It's not changing upon the delivery. It's like you're gonna see like a slightly like really minor changes, and that was what we saw basically. This is like really fast. We are still working on this. We are still trying to understand why the protein are going in, why we are getting signals, and well, which is the reason why the proteins are getting inside the cell. And just to give you some new data, well, confocal data, we are running some experiment with the huntingtin protein, which is another, which is a really important protein actually for the Huntington disease. But what you can see here is basically the confocal experiment that we acquired on neurons, for detached neurons. Still, those are still detached, and you can clearly see how the protein, which was labeled with the ATO48, is more or less all over the place, and you can clearly see where the nucleus is in this case with the DAPI, and that's really interesting because we managed to get those this spectra using an external protein concentration of 100 micromolar, which is pretty low. And at the same time, we did the same experiment, getting less intensity, obviously, using 20 micromolar protein concentration, which is probably saying that there is like a proper biological reason why the protein is going in, and probably this story is not limited by the concentration, or is not depending, is not related to the concentration that you're using. Just to close, and probably I took more time. Just to close everything. So with this in-cell approach, what I think the community is trying to do is basically to push here on this border between proteins and cells. And what we are willing probably to do is to push this equilibrium towards more complex system in order to understand better diseases, or understand how to cure something, or how to contribute from spectroscopies to something that is way more biological. On the future steps and other possible experiment that we are planning to do, or that everyone can try to do, is of course to understand why the shot procedure thermal simulation is working. And we are working on that using some labels and some really biological strange approach. And it would be great to develop like a microfluidic system for doing this. And then obviously the NMR extension is a must, and we are working on that. Hopefully we're gonna get this. We are gonna get that soon. And yeah, well, from the EPR point of view, of course, there are multiple stuff that that can be done, like using orthogonally spin labeled samples, or using the 19F Pandora experiment, which were like beautifully introduced by Marina. And yeah, well, that's mainly it. And I want to just thank everyone for listening, and all the previous worker and chairman here now at the NIH, the Cloud Lab, and the Bax Lab. All right.
Okay, thanks, Francesco.
Really, yeah.
Thank you for this nice talk. And I think now we open the floor for questions. So you can type your question in the chat, but since we are just 25 people, I think you can raise your hand or unmute yourself if you want to ask a question to Francesco.
Yep.
Okay, maybe I start with a very trivial question. That's not all my topic, but you said that when in a CW spectrum, when you record it in vitro, you have one component. When you record it in cell, you have two components. Maybe you said that, but where is the second component coming from? And
well, so what I can say, I think that as far as I got, this second component, where from my point of view is coming from the proper dynamical changings that are induced by the intracellular environment of the cells. And even the first component in the case of in-cell is not the same as the in vitro. So like we know that inside cells, for example, the viscosity is completely different. So even just the proper dynamic, even if the protein is free, assuming that the protein is free, even just like the experiment, it would be different from the buffer condition due to this viscosity. And we know that because, well, there are plenty of work on how the intracellular environment is composed, and like the crowding, and even like non-specific interaction can play a key role here, or specific interaction. And yeah, there are plenty of reasons why you're going to see some changes, and that's probably the interesting story. I can understand that probably CW is not the correct way to understand this. The best way is obviously NMR.
Okay, so you're saying that you use two components just to fit the spectrum, but you don't have
Yeah.
two populations that are different, let's say, but you have
No,
more
well,
like a distribution.
I think that you're gonna have, you do have some like two population of something, or probably even more, right? Because you can even fit more data in there. But the point is that at the state of the art of the story, I think that it would be really difficult to say why, for 100% sure, saying that, okay, this is due to ubiquity, no specific interaction with mitochondria, or that's yeah.
Okay, we have a question from Alberto. You can unmute yourself. Yeah.
Francesco, first of all, many thanks for this very, very interesting talk. I've actually have a question exactly on the slide, and more specifically on the case of Pichia pastoris. Now, comparing the CWPR spectra of ubiquitin and carbonic anhydrase between in vitro and in the cell, it seems to me as if in the cell it were more mobile than in vitro, which sort of like to me looks counterintuitive. Which explanation do you give?
Yeah, well, we explained that on the paper. That's something that we saw mainly in the Pichia pastoris, and that's well, the effect was actually large. The reason why, well, we don't know the proper reason, but we interpret that like saying that the protein is getting chopped, or the protein is getting is like the overall shape of the carbonic anhydrase is changing and is getting more open. And if you say that, it means, well, it makes sense that your overall tumbling time is different, so you can get sharper line. But well, in the paper, I think that one of the main reasons for this is that the protein is getting some strange pathways for protein degradation. And we tested this using, if you go on the paper and if you go on this supporting on the SI, you see that we ran some experiments for using the carbonic anhydrase labeled, and then we use some protease from yeast. And basically what you can see is the same story. Like your line widths are going up, so which means that the protein is of course getting chopped. As specifically, well, that's not something that is specific to the carbonic anhydrase. And if you stop this protease using some inhibitors, you're going to see that your experiment will stay the same. So that's probably the, you know, the really hard, let's say, side of this kind of approaches, because everything is related to the over concentration that you can reach inside cells. If the concentration is low, you're going to see all of this equilibrium that are not saturated. And even like just proteases, they're going to be more, let's say, they can be more active to degrade, to, you know, to chop your protein out. Yeah, that's my let's say our interpretation of this. Yeah.
Perfect, thank you.
Yeah, thank you.
Okay, are there other questions from the audience?
I have maybe a short question.
Yeah, go ahead, Joe.
On the same topic of this two component, did you measure the decay rates of the independent components at all?
Well, in this case, in the case of bacteria and yeast, no. We did that in the case of mammalian cells. And well, we measured the TM of the signal, the T2 relaxation rate.
Oh no, not the. I don't speak of that decay rate. I mean the.
Okay, sorry.
The losing of your nitroxide signal with respect to time over minutes. Did you?
Okay, yeah, you're saying just you say the decay of the signal over time.
Mm-hmm. Yeah.
Yeah, well, we measured that, but we never fit that, because I think that it's really complicated. Well,
Of course,
it would be really easy to get some wrong data there, wrong fitting there. So, but what we are trying to do now, and this is like a little view on the project that we are running now. We are trying to use, in the case of mammalian, the decay as the indication of where the protein is, because in the case of mammalian, some loci, specific loci of the mammalians has different kind of reducting properties. So in principle, you can, let's say, estimate like a ratio between, let's say, the protein is fully cytoplasmatic or is half cytoplasmatic, half nuclear, or something like that. We are still on the starting of this, but it would be interesting to use that stuff for understanding this kind of distribution, even because it will be an application of CW EPR.
Yeah, absolutely. Thank you.
Thank you.
Okay. If there are no more questions, I would close the session here.
All right.
Okay, thanks everybody for coming and tuning in, and thanks Francesco for this
Thanks, guys.
very nice talk. Yeah, and we
Thank
stay in
you.
touch. All right.
Thank you.
Thank you, everyone.
See you all.
Bye bye.